Hey everyone, my name is Daniil Gataulin. I'm one of senior research analysts covering biotech here at Chardon, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. It is my pleasure to introduce Jay Ducker, the CEO of EyePoint Therapeutics, and George Elston, the CFO, to our virtual ophthalmology summit, where the initial focus is on AMD and diabetic-related ocular diseases. Jay, George, welcome.
Thank you very much, Dan.
And Jay, I would like to congratulate you on your new role as a CEO.
Thank you very much. Yeah, it's 2 weeks into the job. Everything's going great.
Excellent. Great to hear. So to start things off, could you provide a brief overview of EyePoint and perhaps highlight some of the recent milestones?
Of course. So EyePoint is a publicly traded company. Our NASDAQ ticker is EYPT. We've been around for a while, and EyePoint is really a drug delivery company, really now concentrating on drug delivery to the back of the eye, to the posterior segment, to the retinal area. So we're focusing on sustained drug release for serious retinal disorders, such as diabetic retinopathy, diabetic macular edema, and of course, wet age-related macular degeneration. We have a proprietary sustained release system that's called Durasert. Durasert has been in 4 FDA-approved products, all of which are now licensed out to other companies. At present, EyePoint does not have any marketed drugs that we commercialized ourselves. Those 4 products all featured the Durasert technology that was not bioerodible. What we're doing now with what we call Durasert E is a bioerodible insert technology, which allows us to deliver anti-VEGF in the form of a small molecule tyrosine kinase inhibitor called vorolanib to the back of the eye for extended periods of time. In fact, we believe with each injection, patients on average should go at least 6 months, hopefully with our drug alone. The drug release can be tailored via different approaches to Durasert to allow for release as short as several months or even as long as 3 years, such as in the YUTIQ insert that we developed. So the Durasert technology is very programmable. It's also, because it's been in, we estimate about 80,000 eyes throughout the years, very safe, really has an excellent safety record. So the pipeline drug that we're developing, we call EYP-1901, that's Durasert E for erodible with vorolanib, the small molecule tyrosine kinase inhibitor. And the recent milestones, again, we've made terrific progress with this. Not just in the last year, but in previous years. I like to highlight the fact that the first patient was treated with EYP-1901 in our phase 1 DAVIO trial in January of 2021. And we will have top-line data from our phase 2 DAVIO 2 trial in wet AMD in December of 2023. So we've made significant progress over the last couple of years. Just to highlight in the last year, however, we have fully enrolled our 2 phase 2 active trials. That's the DAVIO 2 trial in wet age-related macular degeneration. We were oversubscribed with 160 patients fully enrolled. We've also fully enrolled our PAVIA trial, which is for non-proliferative diabetic retinopathy, or NPDR. That trial enrolled 77 patients, also over subscribed. We expect top-line data from the PAVIA trial in the second quarter of next year. I think really importantly for the positioning, the strategy, and the future of the company, a terrific milestone that we achieved in May of this year is we sold our YUTIQ franchise to Alimera. We got $75 million up front. This enabled us to completely retire all our debt, to really improve our balance sheet and give us a cash runway into 2025.
Excellent. Thank you for that detailed overview. Before we jump into some of the data for 1901 and expectations for the program, so I wanted to spend a couple more minutes on Durasert technology itself. Can you highlight which applications it's been approved for, and what changes did you have to make it bioerodible?
Sure.
And I guess another question, I'll add that right away as well, is you mentioned how programmable it is. So I wanted to understand what that means and how do you control those with Duracert technology?
All excellent questions. So the initial Duracert FDA-approved product was Vitrasert, or the ganciclovir implant. It was approved for CMV retinitis in patients who are HIV positive, which at the time of approval in the mid-1990s was a huge problem. In fact, this was my first introduction to the technology. I was one of the original investigators, and I placed over 300 of those implants in patients' eyes and was really impressed with the safety and the reliability of the sustained drug release. In fact, I still have patients in my practice that I still see who have these implants in their eye after 25 years, and the eye sees well with no complications. After that, the company developed a series of corticosteroid implants, one of which was surgically placed, and that implant was for posterior segment uveitis and was licensed to Bausch & Lomb. And then later on, the technology was changed to allow it to be injected in the office as opposed to placed surgically. So the intraoffice inserts, really there are two with that on the market with corticosteroids, both of which Alimera now owns the rights to, one of which is indicated for diabetic macular edema, and the other is for non-infectious posterior segment uveitis. The implants themselves are very similar for those two indications. They're almost identical. So it really makes sense to put them under one commercial umbrella with the Alimera company. Those technologies featured a core matrix of the drug wrapped in a non-erodible polyamide shell. The purpose of the polyamide shell was to limit the diffusion of the drug. And though the drug actually diffused out of these implants through pores at either end. So the implants were able to control drug release by having the number and the size of the pores. Importantly, though, the polyamide was completely inert, does not interact with the human body at all, but is non-erodible. So it never really goes away. But as I said, we've got patients who have multiple of these inserts in their eye without any problems. Quite simply, in Duracert E, what we did is use essentially the same technology to make the drug core matrix, but took away the polyamide shell. There are several reasons for that. The most important of which, with vorolanib, we needed a certain exposure to the tissue. We didn't need to limit it. Also, the solubility of vorolanib suggested that we didn't need to limit it by this polyamide shell. And finally, we didn't want a lot of these shells hanging around in the eye, even though we haven't seen problems with them. If you think about wet AMD as a disease state and the fact that we're able to inject up to 3 inserts, we hope to get a label for every 6 months. You can imagine over the lifetime of a patient, they would receive a lot of inserts, we hope, in which case they might have a lot of these shells residing in the eye if we were to continue to use them. So for all those reasons, we took the shell away, referred to it as Duracert E. Now, there are no new excipients in Duracert E. And so we wouldn't expect any new safety issues to come out of it. And we, frankly, at this point in the human trials and in animal studies, haven't really seen any safety issues at all with either the Duracert technology or vorolanib. So a little bit more about the Duracert E in EYP-1901. The inserts themselves are cylindrical, and they're heavier than water. So when they're injected in the eye and they're injected in the doctor's office, just like any anti-VEGF is delivered, a little bit of topical anesthesia, and then a single injection. These inserts then sink to the bottom of the eye, so they're out of the visual axis. They don't break up into pieces. They don't emulsify. There's no hydrogel in them. There's no PEG in them. There's no PLGA in them. And those have caused potential problems with some other products. So we believe that they should be really safe and well tolerated by patients. And so far, that's what we've seen in the studies. So from the point of view of dosing, I think, again, there's 2 issues around what's in the inserts and how do you deliver them. The dose is important, but what the dose is dependent on is the release of the tissue exposure that you need to get and the release rate that you're going to get in humans. So by increasing the payload or the number of inserts, you could increase the dose and increase the tissue exposure. What you're trying to do again is make the tissue exposure adequate for the disease state, and also make the inserts last approximately as long as what you'd like them to last from a commercial perspective and from the physician's perspective. And so even though we believe the Durasert technology will allow vorolanib to be released for approximately 9 months, we're looking for a label every 6 months for reinjection. And you say, well, why would you do that? The answer is because that's what the doctors want. They want the flexibility to dose up to every 6 months. Doesn't mean every patient will need it that often, but if they have the flexibility on the label, they'll be able to do it. So quite simply, you can, I've alluded to this already, you can alter the dose by the number of inserts. Our current inserts carry a payload of about 1 milligram of vorolanib, and we can inject up to 3 inserts with a single injection, so get to high dose of about 3 milligrams right now. You can change the size of the inserts. You can make them smaller. You can make them larger. You can change the bore of the needle to change the diameter of the inserts. There's also ways we can alter the excipients to make the drug go away faster or slower. But so far, we've been very pleased with the way the inserts have been performed. In our phase 1 trial, we actually studied 2 payloads. One was a 400 microgram insert that was injected through a 25 gauge needle. And we also studied the approximate 1 milligram insert, which is injected through a 22 gauge needle. And we were able to inject up to 3 inserts, again, for the maximum dose of about 3 milligrams.
Got it. I understand. That makes sense. And so the active ingredient in 1901, as you mentioned, is vorolanib, which is a TKI, tyrosine kinase inhibitor. Can you talk a little bit about the mechanism as it and its importance to wet AMD? So,
as you are aware, tyrosine kinase inhibitors are really been around for a while, and they've been really mostly FDA approved for cancer. Some of the tyrosine kinase inhibitors, including vorolanib, have really strong activity against all the VEGF receptors. So that's an important distinction. These molecules work at the receptor level. They're not large molecules that block the ligand, which is what all the FDA-approved products do now. So there's a differentiated mechanism of action here. And in fact, it's possible that there may be some complementary mechanisms of actions going on here with the two different approaches. We haven't shown that yet in a clinical trial, but that's something we're certainly looking for in our phase 2 trial. And more specifically, will we find a subgroup of patients whose vision actually improves under vorolanib treatment in EYP-1901? So it's a small molecule, blocks all isoforms of VEGF. It also has activity against PDGF, and it does not have any activity against the Tie2 receptor at the doses we'd be using clinically. And you say, why is that important? Well, Tie2 is an important receptor in the Ang2 pathway. You may be aware that we now have an approved drug, faricimab, which is now approved in wet AMD and diabetes, and it has blockage of Ang2. If you inactivate Tie2, you activate Ang2, which theoretically would be a bad thing for these diseases to do that. So we have no activity against the Tie2 receptor, which helps to make us feel better about off-target effects. So from the other perspective, again, of why vorolanib and what else does it do for us? First of all, it is patent protected. We've got patent protection out to 2037, and we're going to have some other patents that we're filing along with our new injected system. So we felt very comfortable with patent protection. Vorolanib fits very well into our Durasert. And interestingly, vorolanib had been studied in humans prior to our studies in wet AMD. It was studied by a company called Tyrogenex, who first developed it. It was at the time called X82, and it went through both phase 1 and phase 2 trials as an oral agent for wet macular degeneration. Now, it did pretty well from an efficacy perspective, and there were no ocular side effects, but it unfortunately did have the systemic side effects that these TKIs have, in particular, hepatobiliary toxicity. So it never made it out of phase 2. However, we've got a pretty nice database of safety in about 150 patients who received it orally. Obviously, the FDA has seen that as well. And when we're done with our 2 ongoing phase 2 trials, we'll have a database of another 150 patients from a safety and efficacy perspective in EYP-1901. So we'll have a very robust safety record for vorolanib in humans with those 2 databases. Finally, vorolanib may have some other benefits that the large molecules don't have. For example, at ARVO this year, we presented data that an animal model that shows vorolanib may actually be neuroprotective. Neuroprotection means, obviously, in retinal diseases, you may be able to preserve the photoreceptors. If that's the case, then perhaps we will show in at least a subgroup of patients some improvement in visual acuity in eyes treated with EYP-1901. Vorolanib also has activity against PDGF, as I mentioned, and that modulates fibrosis. So by blocking PDGF, we may be able to reduce the fibrosis in these wet AMD lesions. Again, hopefully what we'll be able to show in the longer term would be better visual outcome by doing that. So vorolanib is really a really terrific partner for Durasert in all those ways. And because of all that data, we're optimistic that we will show similar benefits in our phase 2 that we showed in our phase 1.
Got it. So I guess one of the more important aspects, as you mentioned previously, is 6+ months of duration is 1901. But given newer biologics coming to market and in development with longer treatment intervals, what do you see as the value add for 1901? I know you highlighted some of the other benefits, but if you could summarize them here as well.
Well, I think the first benefit is that if our data holds from the phase 1, we got over half the patients out up to 6 months with a single injection. None of the new biologics can do that. They can't go that long. So there is some evidence from the pivotal trials for both Vabysmo, which is currently FDA approved, and high-dose Eylea, which is not yet FDA approved, that they might be able to take out patients longer than Lucentis or standard Eylea. And while that does appear to be true, it appears that the improvement is incremental. In other words, if I've got a wet AMD patient that I'm treating with Eylea every month, and if I try to treat and extend them out to 5 weeks or 6 weeks, I get fluid. So I know they need monthly injections. I switch them to Vabysmo. Chances are I may get them out another week or 2, but I'm not going to be able to jump them to 4 months. That patient has just got too much VEGF activity. On the other hand, we had patients in our phase 1 who were being treated monthly with standard of care anti-VEGF, and we were able to get them out 6 months or longer with a single injection. So that's really the benefit. And when you really think about it, drill down, no matter how much drug you're putting in the eye with a high-dose biologic, after the first day or 2, you're on the downslope of that dosing. And in weeks, that drug is out of the eye. Our drug's not out of the eye because it's consistently being microdosed every day, every minute, every hour for about 9 months. And we think that approach, the zero-order kinetics approach, will show benefit because we will be able to take, we hope, at least 50% out of the patients out 6 months or longer with this approach. So again, these are huge companies that are marketing these drugs. We're well aware that we're up against some of the best companies in the world, but we can do something they can't, which is reliably, and we hope at least half the population go out 6 months or longer.
Got it. So the W2, the phase 2 trial, it's fully enrolled, actually over-enrolled. That's ongoing now. Can you walk us through the trial design and
Absolutely.
Specifically And yeah, and specifically, I want to hear about key learnings from the phase one study that you implemented into phase two that you, in your view, increased the probability of success.
Sure. So just to review the phase one study for a moment, that was called the DAVIO trial. It was an open label dose escalation, no control, no reinjection. It was for maintenance therapy, which means it only enrolled patients that had previously treated wet AMD. 17 eyes. I alluded to some of the results already. About half the patients, a little over half the patients were able to go out up to 6 months or longer. Stable visual acuities. The visual acuity in this group dropped about 2.5 letters in 6 months, a little over 4 letters in 1 year with no reinjection. No safety issues at all. Very clean safety record. But what we learned is eyes that were not doing well with standard of care, meaning they got an Eylea injection and then a month later they had more fluid, we didn't have much of a benefit for them. The truth is there's not a lot of patients out there in the real world like that. Most patients respond, at least anatomically. So if you ask a retina specialist, well, how many times will you have a patient under Eylea treatment or Vabysmo treatment, and you'd give them a monthly injection, and they come back in a month and they've got more fluid? And you'll say, well, I hardly ever see that. Well, we had 17% in the phase one because that's the type of patient that you get enrolled in a phase one. It's the bad patients, not the good patients. But the learning was we're going to try to eliminate those patients from the phase two. And so the way we set up the phase two, everybody needed to get a standard of care injection of an anti-VEGF, didn't matter which one, between 2 weeks and 5 weeks prior to screening. And at screening, they had to have 350 microns or less of intraretinal fluid. It means they could have, CSD, complete fluid on OCT. It means they could have some fluid. We're not requiring them to be completely dry, but they, after a recent injection of a Lucentis or Avastin or Eylea, if they had more than 350 microns of fluid, it means they weren't really responding. So that's one learning that we applied to the phase two. The second learning from the phase one is kind of the opposite of the bad eyes. The good eyes did really well in the phase one. If you want to say what's a good eye is an eye that even if they're getting monthly therapy, they're completely dry. Now, their eyes like that, they're not failing anti-VEGFs. In fact, they're really responding. They just can't go very long between the shots. And eyes that entered the phase one completely dry did exceptionally well. 92% reduction in treatment burden at 6 months, 89% at a year, 67% were rescue-free up to 6 months. Over 50% of that group was rescue-free up to a year. So the message there was, if you're responding really well to an anti-VEGF, you're probably going to do really well with the EYP-1901. So what do? Even though this is maintenance therapy, all the patients in the trial got re-induced with Eylea. They got monthly injections ×3. So the way the study was set up, randomized trial, 3 groups. All the patients on day 1 got an Eylea shot. On month 1, they got an Eylea shot. On month 2, they got an Eylea shot. Then they either got a sham 1901 or they got a real 1901. And the real 1901 was either 2 milligrams or 3 milligrams. And then they were seen monthly. Every 2 months, though, they either got a sham Eylea or they got a real Eylea, depending on which group they were in. Primary outcome is non-inferior visual acuity change against the Eylea control. And as the FDA has declared publicly, the non-inferiority margin is 4.5 letters. So that's the primary outcome that we're looking for, non-inferiority, although this study was not powered to show a statistical non-inferiority. Obviously, safety is still really critical in this field, as we keep seeing over and over. So we hope to see a similar safety profile as we did in the phase one. And then the other big thing to look for, I think, with the phase 2 results would be a significant reduction in the treatment burden. In most other studies, and in our phase 1, the way treatment burden was measured is retrospectively, you looked at what did they get leading up to their enrollment, how many shots on average, and then how many shots did they get after enrollment? And then you compared the 2 figures. And as people have pointed out, that's not totally accurate because what they got leading into the study wasn't part of the study. So the beauty of what we're able to do is we're going to be able to have prospective treatment burden measurement. We know what the Eylea arm is going to get over the 6 months after the 1901 goes in. On average, it should be about 3 injections every other month. Although I will point out the Eylea arm can get rescued in this study too. So you might actually see a little bit more than 3, but 3 injections is what we expect. If we do as well as we did in the phase 1, we had 1.2 injections after 1901 went in the phase 1 in the first 6 months. So 1.2 divided by 3, that'd be a 60-some% reduction in treatment burden. That would be an excellent result, because we think we could, even we don't have to do quite that well to have a commercially acceptable drug. So for us, those are the 3 big outcomes we're looking for. We'll also have other subgroup analyses in the phase 2. And as I said already, we think through inclusion-exclusion criteria and through the load of everyone, we have reason to believe that we may do better in the phase 2 than we did in the phase 1.
Excellent. And speaking of rescue injections, what are the rescue or retreatment criteria in this trial, and how do they differ from other programs?
Yeah, it's a good question. So in the Davio phase 1, our rescue criteria, I'd say, were the standard phase 1. Drop in 10 letters from the baseline due to wet AMD. 10 letters is 2 lines on the eye chart. New fluid of 75 microns due to wet AMD, new hemorrhage due to wet AMD, or at the investigator's discretion. We changed that a little in Davio 2. So the visual acuity now is a drop of 5 letters or 1 line, but it has to be associated with 75 microns of new fluid, and it's against best in study, not necessarily at screening. New fluid of 100 microns from best on study, but it had to be 2 visits in a row. And the reason we did that is if the vision is stable, but you've got some new fluid, but you have a sustained release device releasing medicine, that new fluid may go away. We saw that in our phase 1. And Genentech saw it in their port delivery system studies as well. Sustained release eyes actually may act a little different. New hemorrhage is in there as well. And of course, investigators' discretion is in there also. I do want to say something about the rescue retreatment criteria, actually a couple things. First of all, it doesn't seem that the FDA has any tight parameters around that. And it's hard to compare study to study because, as I think you're alluding to, each study can be different. And I think what's interesting is the closest study to what we're trying to do would be Genentech's port delivery. And that, because it's maintenance therapy, just like what we're trying to do. And obviously there's some differences, but their phase 3 rescue criteria was very different than their phase 2. And in some cases, they allowed up to 150 microns of new fluid. So it appears, that the FDA has some gives companies leeway around that rescue criteria, and it's not a strict thing by the FDA. The other point I'd like to make is when you're trying to compare rescue criteria in a study like ours or port delivery, you can't really compare it to the Vabysmo and to the high-dose Eylea phase 3, because those criteria are not rescue criteria. They were criteria to move a patient into a different treatment interval. So that was applied when a patient showed up for a shot anyway. So they were getting a shot on that day. And if they had a certain criteria, they were moved to a shorter interval. So that's not really rescue per se. It was criteria for changing the interval, which is not exactly the same. Also, just makes it a little bit harder to compare apples to oranges.
Got it. Makes sense. And, you have a phase 2 readout in December, I believe. What are you seeing as I guess as an ideal case scenario, and what do you anticipate your next steps for a wet AMD program to be?
So I think I alluded to the 3 main things we're looking for. Obviously, non-inferior visual acuity, and what we're looking for there is a number that, if in a large enough study, would be statistically non-inferior to the Eylea control. So we don't know what that number might be, but I can tell you if 4.5 is the goal, it's theoretically possible that you could be 4 letters worse than Eylea and be statistically non-inferior if the standard deviation of the change in visual acuity is small enough. Now, we don't really know what the change in visual acuity standard deviation is going to be. That's one of the reasons we ran the phase 2. So that's the kind of number we're looking for. And obviously, the closer we are to Eylea, the better. And I wouldn't say we expect it, but is it possible that we could be better than Eylea? I think it's certainly possible. And so from a, kind of what's the base case, what's the best case? I think obviously best case would be we'd be numerically superior to Eylea. From a safety perspective, again, There's not a lot of margin for error with safety in the retinal field, as we've learned. The good news is we didn't have any safety issues in the phase 1. We don't expect to see any. Vorolanib appears to be very safe both in animals and in humans, and we've tested it in animal doses 10 times higher than we could ever put into a human with no toxicity. Our inserts appear to be safe. We've tested up to 6 inserts into a rabbit eye, which is about the quarter size of a human eye, with no safety issues. And so we're, anticipating a good safety outcome here, but anything can happen in a human. And finally, reduction in treatment burden. Again, that's the benefit to the patient. How does the patient benefit from this? And again, I think we talked a little bit about that. If we even have a 40% or 50% reduction in treatment burden, that's a benefit to the patient because you can cut their visits in half.
Yes, got it. I wanted to shift gears and talk a little bit about NPDR. First, one of the main differences is the treatment or treatment paradigm. Very few patients do actually receive active treatment for NPDR. So my question is, why do you think they will be more open to receiving 1901? And maybe in that answer, you can incorporate some of the patient or physician feedback as you're thinking about the market opportunity for NPDR.
Yeah, so the answer is simple. It's timing the injections interval that's necessary to treat the disease. So most NPDR patients have minimal symptoms. They're young. They're usually working. And most of the time, they're being seen by their eye care specialist or retina specialist once a year, once every 9 months, maybe once every 6 months. Then if you ask them to come in monthly for an eye injection, and then every quarter or so after that for an eye injection, that's a big burden on their lives. They're already seeing their doctors a lot because they're diabetics. And if they don't perceive that they're getting benefit from it, I think it's hard for the doctor to prescribe it. It's hard for the patient to agree to it. On the other hand, if you have an injection that can be delivered every 9 months or even once a year and gives a similar type benefit with a similar safety profile, that doesn't change their visit paradigm at all. Come in once a year, well, now I've got a shot I can give you, and it will reduce your chances of having, a significant complication from diabetic retinopathy that may be eventually treatable, but would require multiple injections. I think that's something that will definitely appeal to patients. And we saw it in the Pavia trial with the recruitment. It was not a difficult trial to recruit to because we were only doing 1 injection over 9 months or sham. And so we think that that's a real open market right now because the current ligand blockers have been out there for a while and haven't really made a dent in it, most likely because of the frequency requirements.
Got it. So you did mention that you already recruited that phase 2 trial, and again, similar to WO2, you oversubscribed. Mm-hmm. Can you talk a little bit about that study design and
Sure.
key differences from the wet AMD trial?
So one of the key differences for all of us studying NPDR is, at least as of now, the FDA does not require an active control arm. Even though there are 2 medicines that are FDA approved, Lucentis and Eylea, you're able to do a sham injection as control. And the reason is standard of care is observation right now. So that makes the study easier to recruit to, especially if you're only doing an injection every 9 months, not every month or every quarter, and reduces the cost of the study and probably reduces the N of the study also. So for a diabetic indication, if you have a choice of doing DME or NPDR made more sense to us as the second indication. We're certainly going to do DME also. It's a big market, but NPDR made more sense for those reasons that I stated. Easier to recruit, faster study, cheaper study, and potentially a huge unmet need out there that we may be able to tap into that the others weren't able to.
Okay, that makes sense. And you also mentioned that you are planning to go into DME. Mm-hmm. Can you comment on the potential timelines and what you want to see from wet AMD or NPDR before going into DME?
Well, we're going to start DME in the first quarter of next year. So we will have the top line wet AMD data at that point. Every anti-VEGF that's worked in one indication has worked in all indications. to at least a varying degree. So if our top line data from wet AMD is good, we're obviously going to be full speed ahead on DME. And we would expect the phase 2 trial to be a small proof of concept trial, which again, if positive, would allow us to go right to, at that point, we hope 1 pivotal in DME. At least for now, that would be our plan: 2 pivotals in wet AMD, 2 pivotals in NPDR, and then a single pivotal in DME, and other indications after that, RVO, myopic CNV, other things that we might consider getting a label for.
That makes sense. And in the last couple of minutes, I have a couple more quick questions. One, in terms of manufacturing and EyePoint manufacturing capabilities, what does the company offer in terms of supporting both clinical and potentially commercial supply?
Sure, it's a great question. First of all, I want to say that all manufacture of YUTIQ in EYP-1901 is currently done in our Watertown facility. And we are still manufacturing YUTIQ. We're currently manufacturing for not only Alimera here in the United States, but we're manufacturing for our Chinese partner, Ocumension. And we do expect for the foreseeable future to continue to manufacture YUTIQ. We have enough manufacturing capability to manufacture the clinical supply for the phase 3 trials in Watertown. We don't expect to tech transfer this really ever. There's a lot of know-how involved in the manufacture of Durasert E, as well as non-erodible Durasert, and we intend to keep this manufacturing capability in-house. So in saying that, we did sign a lease and have broken ground, and actually at this point even poured concrete on a modern manufacturing facility in Northbridge, Massachusetts. Northbridge is about 50-minute drive west of our headquarters in Watertown. We have a great partner out there. It's a great leasing arrangement for us. And once that facility is up and running, we should be able to manufacture up to 1 million doses or more of EYP-1901 to fully take care of the potential commercial needs. From a time perspective, and George, correct me if I'm wrong here, I think we've got about a year of construction to do. Then it takes about 6 months more to get the FDA to approve the qualifying lots and do their inspection. So we think that facility will probably be up and running in about approximately 1.5 years from now.
Yeah, and the plan will be to utilize it for obviously our phase 3 trials and registration batches for the NDA out of that commercial facility.
Got it. Excellent. And George, another question for you. Could you remind us what the company's cash position currently is, and where do you expect the runway to carry you through?
Yeah, so as Jay mentioned, we did transact the YUTIQ franchise in May, and that was an $82.5 million transaction plus future royalties received. We received $75 million upfront as part of that, and we were able to remove our existing bank debt obligations. So the net to our balance sheet was a little over $40 million, and we ended Q2 2023 with about $142.5 million. We also extended our cash runway guidance into 2025, and that assumes that the continued phase 2 trials for wet AMD and NPDR, and also the initiation of the phase 2 in DME next year. So our balance sheet's in a great place. We've got a great cash position. Debt is gone, and we're well funded through into and through key milestones in the next year.
Excellent. And one last question, Jay, maybe in a minute or so. What do you think the investors are still missing about EyePoint's story?
So I think the main thing that's being missed here is the new potential to carve out a paradigm shift in wet AMD therapy. I think some investors are looking at us as going head-to-head against high-dose Eylea and Vabysmo, and that's just really not the case. First of all, we're not going to be induction therapy. We're going to be maintenance therapy. And secondly, there may be some patients that still require some of the treatment with those ligand blockers. So I think that's the main thing they're missing, is we're not trying to replace them. we were complementary to them. And we believe that our aspects of our drug, again, if the phase 1 data holds, I think that we are likely have an approvable drug, and I think we likely have a very a drug that's very likely to be successful in the marketplace. Now, taking a look at our stock price over the last year, I think some investors are starting to recognize that we do have this data coming out in December. We have a very good looking balance sheet at this point. And I think that we have the potential to have a drug that would be a multi-billion-dollar a year drug. So I think the word is starting to filter out that this is an exciting opportunity.
Excellent. Well, we are out of time now, but again, thank you for joining us for this summit. I really appreciate your time, Jay and George. Great to see you as always.
Thank you very much.